So I've already mentioned that next-gen sequencing generates a lot of errors. So it's estimated that it has a 10 times higher error rate than traditional Sanger sequencing. Now these errors are annoying, especially when you want to do variant calling, for instance, but in all applications it's annoying, but especially there, these errors are very annoying. Because it's very hard to make a distinction between a variant, which is a different base than in the reference, and a sequencing error, which is a different base than in the reference. So you need to have a way to make a distinction between sequencing errors and real mutations, real variants. So what is the solution to be able to do this? The solution is to sequence a lot, so that every part of the genome or every part of the transcriptome for RNA-seq data is covered in multiple reads. And this is what you see here in this example. So this is the reference sequence here, and these are your reads. And you see, for instance, that this part of the reference is covered in all these reads. And that's what they call coverage. It's the average number of times that every base in the reference sequence is covered in the reads. So for this example here, the coverage varies between 5 and 7. Yeah, you will have parts of the reference where there are only 5 reads, and you will have parts where you have 7 reads. So the coverage can vary over the genome. You will have regions that are easy to sequence with higher coverage, and you will have regions that are hard to sequence with low coverage. So this is something that is important, and when you report the results of your sequencing, that is a factor that you report. What is the coverage in your sequencing experiment? And why is this important to have high coverage? Well, it allows you to make the distinction between real variations and sequencing errors. So what is a sequencing error? A sequencing error is a variation, a mismatch with the reference that only occurs once. Because these sequencing errors, they occur randomly. So you don't expect them to occur twice in the same location. Yeah. A real variation can then be found because it occurs multiple times in your reads. So for these data, you could say here, this is a sequencing error and this will be a real variation, a real mutation in the genome. So coverage is important, and we always get questions about what is the coverage that I need for my experiment. Well, first of all, something very important. Coverage, this word here, can have different meanings depending on who you talk to. So a bioinformatician will use this definition. So he will say the coverage here is 5 to 7 because each base is represented 5 to 7 times in your reads here, each base of the reference. But a sequence provider, when he talks about coverage, he just means what is the total number of reads that you want to generate for each sample. But that's a different number. This is millions of reads, what a sequence provider calls coverage. How can you calculate what you need here? So there are formulas that allow you to calculate the coverage that you have to tell to your sequence provider. And that's what you see here. So for DNA-seq, you say, well, it's dependent, of course, on the genome size, it's dependent on the length of your reads, and it's dependent on how much you want here, yeah? What the coverage is that you want here. So for variant analysis, you want a lot of coverage here. So you take a high number, you have to multiply it with the genome size, all the bases in the genome, and you have to divide by the read length. And that number that you get here as a result will be the coverage for the sequence provider. The number of millions of reads that you need. For RNA-seq, you do the same thing, but of course for RNA-seq, you're not looking at the full genome. You're only looking at the genes. And some of the genes will be off, they will not be expressed, but you don't know which genes are not expressed. So the best solution that you can do here is instead of the genome size, you take the exome size. So the total number of bases that are covered in exons. And it will not be accurate because some of the exons will be not expressed at all, but it's the best you can get here. And why is it important to tell your sequence provider which coverage that you need? Well, your sequence provider will use that number to see which sequencer he has to use. You have sequencers where you can generate a lot of reads, and you have sequencers where you can't generate that many reads. So depending on what you want, he has to use a different sequencer. So he has to make sure that there's not too much clusters on the flow cell. That's basically why this is important. Okay, so that still hasn't answered your question, what coverage do I need? I've shown how to calculate it, but you still need to choose something here. Yeah? A number. What number do you choose here? Well, that depends on the application, and there's not a perfect answer for that. Nobody knows what exactly you need. Basically, the easiest rule of thumb is you choose the coverage that you can pay for. The higher the coverage, the more reads, the more expensive the sequencing will get. Also important to realize, high coverage is not a miracle solution. If there are fragments missing in your library, you cannot make them appear again by having high coverage. If your library sucks, then nothing can save it, even not high coverage. Okay, so the numbers. So the numbers depend on which application you're going to do. So you can immediately see that high coverage will be important when you want to do variant analysis. So DNA-seq for finding mutations. But even there, it depends on what you exactly want to do. Two examples here. So you have one patient, but for that patient, you want to be 100% sure whether he has a certain mutation or not. In that case, you need to go for very high coverage because you really want to know if this is a real mutation, yes or no. So the higher the coverage, the more sure you will be. What is high coverage? 30. 30. So 30 reads on average per base in your reference. That's high coverage. Now, alternatively, there's another experimental setup possible here. You look at the whole population and you want to look at mutations in the population. Which mutations occur in the populations and at which frequency and that kind of thing. So an example, the 1000 Genomes Project, where they sequenced more than 1000 people to just look at this. What are all the variations in the human population? So here you have more than 1000 people, so you have more than 1000 samples. Yeah? You cannot go for 30, yeah? That would be really expensive. So nobody can do that. So for these kind of experiments, they take, the 1000 Genomes Project took a coverage of 4 here. So 4 reads on average per base in the genome. That means that for every individual, you will not be 100% sure if that individual has a mutation or not because you only have a coverage of 4. But on the population level, since you have 1000 individuals, you can still see if a variation is real or not. If multiple people have it, well, then it's real. So this is how you solve it in that case. So for variant analysis, I also asked the nucleomics score, that's the VIB sequence provider. So they do sequencing the whole day, every week, every year. I asked them what they recommend to their customers. And You can see their answer here. Not going to read it out loud, you can read it yourself. So what I want to mention here is that for cancer samples you need real high coverage, 100. And the reason for that is that cancer samples are never pure. So you will have a bit of tumor, but you will also have normal tissue in your samples. And that dilutes your tumor signals. If you want to find mutations in your tumor, you need to have real high coverage here. That's why it's so high. So what they say here, that if you want to call variants, the tools that do this, GATK, that's the tool that calls variants, needs at least a coverage of 8 before it will call variants. And that's a coverage of 8 being 8 reads that show a mutation. And then GATK will believe it. So it means that you need a coverage that's even higher because not every read might show the mutation. Yeah? So that's why they say at least 20. Then we can work with this threshold in GATK. For ChIP-seq and ATAC-seq, this is what they recommend. So these are numbers. They are a sequence provider, so if they talk about coverage, they often express it in millions of reads. So that's what they say here. For ChIP-seq, it's normal that you will have different coverage for your ChIP sample. That's a very small library because it's only the reads, only the fragments that bind to the transcription factor, and your control sample, because this is full genomic DNA cut into pieces. So the genomic sample will be bigger than the ChIP sample. So because the ChIP sample is so small, you can increase the coverage. So typically the ChIP sample will have more coverage, but not in total number of reads, but I mean more coverage in this number. In total number, the control sample will probably be bigger. And then for RNA-seq, this is what they recommend for differential expression analysis. So here it also depends on how good your DNA, your RNA is. If it's degraded, you need more, obviously. Yeah? For RNA-seq, it's very important that you don't go for very high coverage. It's not necessary. Yeah? You're not going to call variants, so you don't need to have high coverage. It's much better for RNA-seq to go for more biological replicates and low coverage. Unless you want to do the specific applications like looking at mutations, splice variants, looking at variation, or looking at fusions, which are also a form of variation. When you want to look at variation, again, you're here in this case, then it becomes important to have high coverage. So here also, when you want to look at mutations, the coverage increases.